study. And similarly, the thyroid, 35%, lifetime risk, endometrial and renal. I would say those are in the 25 to 35% range. And colon cancer, to our shock, because before 2010, we didn't know that. And it's twice, 9% is about twice the risk of the general population. And melanoma, which we always suspected, never thought we could prove it, it's 6% because it's common enough in the general population. So because we know when the age in which the risk rises, we can sort of say, well, it would be smart to start screening right before that. And so take the breast, for example. We start screening around age 30, and now we know it's not considered MRI, breast MRI for sure, and mammograms. I'm not going to go in detail because the expert subspecialists are here, and they will address it in great detail. So we asked, if you get the first cancer, what is the second cancer risk? So we took a 7-year multi-institutional study, ours, meaning we had patients come into our registry, and there were, and I said that cancer is mostly adult onset, so we only looked at the adult patients. And of the almost 3,000, there were 2,000 different invasive cancers. And everyone had their PTEN encyclopedias read, and 114 had a disease-associated PTEN mutation. We always look at the objective pathology records and medical records, and then we compared it to what we consider is the general population, which is the SEER registry. And when you make that comparison, it's called the standardized incidence ratio, SIR, of a second malignant neoplasm. So it's not a recurrence, it's a second primary cancer, SMN. So to summarize, of 114 adult patients, 46 or 40% had SMN. We quickly looked at it again before I came, and this number is holding, it's 46%. And the median age at that second primary cancer is 50, with a pretty broad range. The median interval between that primary cancer and the second one is 5 years. Usually when it's very short, at less than 1 year, and it's the same cancer, so for example, the first cancer is breast, the second is breast, we look very carefully that it's not a recurrence but a second primary. And here is the summary. Just ask you to look at the top one. So the SMN risk for all cancers after a first cancer is 7-fold that of the SEER registry in the general population. And this is just in graphical form. Again, x-axis is age, y-axis is fraction, so percentage. And at the top is, if there is a first cancer, the highest curve, the black one, is that breast cancer risk, followed by endometrial, which is interesting. So what are the take-home messages? Individuals with a germline PTEN mutation are at increased risk for second malignant neoplasms as well, compared to the general population. And so lifelong enhanced surveillance is necessary. Some have advocated prophylactic mastectomy, given the high risk of primary and secondary. I hope Dr. Grobmeier will address that. And we are following up our cohort, so it's been several years since then. We need to increase sample size. And it's very important to see a validation by another group. We haven't seen that yet. And to summarize facts, knowledge is power. We know that PTEN germline mutation predisposes to breast, female breast, thyroid, uterine, kidney, and colorectal cancers, and melanoma. Therefore, because of that knowledge, we're able to bring in the PTEN-informed multidisciplinary care, which is summarized here. Usually it starts with a genetics evaluation. We take your history, we take the family history, we consider the diagnosis, and then there's genetic counseling. It's PTEN-informed genetic counseling, the blood draw to test for the gene, and if the results are positive, we know what the risks are, and so the multi-care team, many of whom are here, will look after you. So this is our PTEN Cowden Clinic website with contacts. And then we want to speak about research because, as you can see, the research over the last 2 decades has informed clinical care. And so this is our original study, what we call 8458. So anyone with a known PTEN mutation or a variation of unknown significance, which is actually very rare in PTEN, or anyone meeting the complex international counting consortium criteria. If you're wondering, Shreya will help you. And more recently, there's a little algorithms on our website. You can even type in PTEN score. Anyone with a PTEN risk score of 10 or higher will be eligible, and that's our website. And Dr. Harden, Dr. Sid are here, and this is the U54 study, which is looking at autism and PTEN. This is the longitudinal study, so we follow you for 2 months and just remind everyone that PTEN is involved with autism and macrocephaly of about 10%. And in the longitudinal study, we're looking at a snapshot, so cross-sectional, on neurobehavioral markers, as well as longitudinal. Some of you have come time 0, year 1, and year 2, and comparing the cognitive and the symptoms. And a secondary endpoint, because many are kids and young adults, is there cancer that occurs during follow-up. We then also collected blood, and so we'll say, as well as neurocognitive testing. And so we go, are there any cognitive markers associated with various scores or symptoms? And there's an MRI. Are there imaging markers? And finally, my lab has also looked at the blood and say, are there blood markers of the brain? This is just a little advertisement that we have a pilot randomized controlled trial of an mTOR inhibitor, so everolimus. So PTEN is up here. It talks to the next molecule and the next protein, and mTOR is down here. When PTEN is altered, misspelled, mTOR goes way up, and so an mTOR inhibitor to push it down is what we have. And as individuals with ASD and PTEN mutations, that's just the rationale that actually Dr. Fraser put together for me of why it is rational to go into this clinical trial. And so the trial is now open. We do randomize the subjects to drug and no drug. It's important to know because many of our patients says, but I don't want to get the placebo. Everyone on placebo will cross over to the drug after 6 months. And so we do need patients, and Dr. Sid will probably address this. The U54 group, the clinical trials group, and Dr. Harden is the leader, we are going to relax the criteria of age. So it was 6 to 21, and we're going from 5 years old to 45 years old. And with that, let me end and catch up in time so that we can give the other speakers time. I want to thank my team. I did not do it alone, never have, over the last 22 years. And the work I showed you is mainly that of Dr. Stan and Yao. And these are our physicians in the Center for Personalized Genetic Healthcare. Jessie Mester, whom you all love, who is on our medical board, and Shreya now. And I'm glad she could come so she could meet all of you and you can meet her. And this is our multi-care team, many of whom are here. Unfortunately, our dermatologist is not here, and our GY and onc, but I think you met them last night at the reception. Dr. Berg will be rushing in from the airport to give her talk, and you will hear from all of them, and I thank them for being with us, looking after all of you. This is our international consortium who helped recruit people from around the world. Thank them as well. Yes, funding is extremely difficult to get, and so we thank our funders. And Dr. Niao is here, and I wanted to show that. I know Toys R Us are going bust, but PTEN R Us are not. And this is my lab in 2017. This is more recently. You notice that Dr. Bush was here, Dr. Fraser. They're so kind to come and attend our lab meetings, so I make them sort of honorary members of my lab. And I enjoy it very much. But most importantly, thank you for participating in our PTEN research. Thank you for ensuring knowledge is power. So I ended early. Hopefully I didn't go too fast, which means I think there are 5 minutes for questions, because we certainly want time for Dr. Sid to come up after me. I hope I haven't stunned you in silence. All right, very good. I've caught up in time.
Than you have listed. Is there like certain symptoms that you look for to start testing earlier, or in general do you just encourage people to start testing at the ages that were on your PowerPoint?
So the question is, some of the other groups, support groups, or I think even physicians, will start screening at different ages, sometimes earlier than what we have. And I should say that it's because of our work that actually has been replicated by 2 other international groups that forms the NCCN guidelines. So the guidelines don't just pool, it's evidence-based. So that's just the preamble. I think that might be suffering from not keeping up to date. I'll give you a very good example. So for example, before our 2010 study, we said that there's no colon cancer risk. They're just hamartomas like everywhere else. And with our 2010 study, we showed the 2-fold increased risk of colon cancer. We showed that the colon cancer risk didn't occur until the 30s, and that's where we recommend our colonoscopies. Now, before that, there's either no colon cancer risk, or we thought that it occurred much earlier. And so some groups will say, begin colonoscopy in your teens or even at 10. I think you're sort of referring to that. And so without symptoms, I always say that. So kids without symptoms shouldn't have a colonoscopy. If they're bleeding from below or anything GI, of course. Anyone else? Oh, there's 1, 2. All right, see, you've encouraged questions. Good for you.
Okay, who had a question?
Good morning, Dr. Eng. Thank you very much. My question, you mentioned 2 different studies. Are both of those based in Cleveland, or do you have study sites around the country where patients can enroll?
Right, so directly to the Cleveland study. So that's the 8458, the original study. I think you're asking the U54, the autism and PTEN study. That you have to physically come to one of the sites, and there are multiple sites. So Boston, Dr. Sid's represented here. Stanford, Dr. Harden, there he is. Well, you're representing UCLA too, because you carried the 2 plaques. So UCLA couldn't come, and us. And same with the clinical trial. So even if you're a patient living in Hawaii, let's say, or San Diego, we'll say, go to Dr. Harden, or if you're outpatient and living in New Hampshire, we'll go also to Boston University with Dr.
Hey. So do we get feedback from the 3 years we've participated in the study? Like, do we get information on what you find? Do we have to wait on that? Like, what?
Part of the study, well, yes, part of the study, we will look at PTEN mutations again. I mean, although many of you, most should have a PTEN mutation. So if we find something, we will return the results through your center. So let's say you enroll with Dr. Harden, we will give the results back to Dr. Harden, who will tell you, and to say you need to confirm that typographical error, that mutation, in a clinical lab, because remember, my lab's a research lab. Now, in theory, we are always supposed to give back results en bloc. So for example, I can say, so many people X of Y with the autism versus the no autism group showed this or that. But for that, we do have to have the whole cohort because sample size is power versus having just a partial sample size and trying to make a conclusion. And I have to say, research groups, academic groups, we suffer from lack of manpower. So we have not been very good at, having a newsletter made or mailed. I mean, you might recall Jesse Metz has been talking about making a newsletter for years. And we've just been so busy looking after our patients and doing our research and all the regulatory paperwork. Now, when patients come in, we tell them, when the cancer report came out, we in fact sent an email to the cancer report to the research participants and the caregivers who enrolled them. I hope that Kristen's website will be an outlet where you summarize, you really have summarized our research results. Like that would be the more standard and more easy way of getting results on that. Very good.
Thank you, Dr. Eng. Ooh, that's loud. Sorry. Let me step back. A couple of things before we get moving forward. There is travel stipends out in the lobby if you'd like to participate in that. And who all in here is on Twitter? Raise your hand. If you will take your phone out and tweet hashtag PTEN Patient Symposium 2018, Kristen would be so forever grateful. All right, as we move forward, we're going to update on the DSC. Dr. Antonio Hardin is Chief of the Child and Adolescent Psychiatry Division and the Director of the Autism and Developmental Disorders Clinic at Stanford University. His clinical expertise is in the evaluation and treatment of psychiatric disorders in individuals with neurogenetic disorders and/or intellectual disabilities. He has served as a principal investigator on numerous projects examining the neurobiology of autism and assessing the effectiveness of a wide range of interventions in the treatment of developmental disorders. He has led clinical trials examining the usefulness of innovative compounds and novel behavior interventions in the treatment of children and adolescents with autism and/or intellectual disabilities. He has published extensively in the field of developmental disorders with more than 100 peer-reviewed papers. So please welcome Dr. Hardin. Okay. We'll just see which one is it. Is that good? Oh, you should be fine.
Okay. Good morning. Can you hear me? Very good. Okay. Thank you for the invitation. First time in Alabama. I've been around the country, but that's the first time in Alabama. Glad to be here. Thank you, Kristen, for the invitation, and thank you for Dr. Eng and Dr. Frazier for getting me into this field. My background is in neurodevelopmental disabilities and autism, but I've been doing a lot of work in clinical trials, and that's how I got involved. I will talk a little bit about the clinical trial, and then Dr. Sid will come at the end to discuss a little bit some of the findings from the observational studies. So what were the objectives of the clinical trial? The first thing that we focus on is on the safety of any agent that we're trying to use. So that was the primary aim of what we were trying to look at the study, to see if everolimus, which is an mTOR inhibitor, we'll talk a little bit briefly about it, and Dr. Eng covered a little bit some of the information about it. So that's the first thing we look at, the safety of this drug in individuals with PTEN mutation. Everolimus has been studied in a wide range of patient population, including kids, adolescents, and adults. But we don't know how this molecule, how this compound is going to interact or react in individual with PTEN mutation. So that's why that's the main focus of the trial. However, there's a lot of secondary objectives. One of the main secondary objectives to see if everolimus is effective in targeting neurocognitive deficits in individuals with PTEN mutation. And in addition to that, we wanted to look to see at other areas of potential benefits. Behavioral benefits, mood dysregulation, autism symptomatology, cognitive functioning, and adaptive abilities. In addition to these secondary measures, we're also looking at some biomeasures, some more objective that are biologically based, such as eye tracking tasks, resting state EEG, electroencephalography, and auditory evoked potential. So these are indirect way of measuring some of the effect of the medication. Now, one of the exploratory aims that we are aiming to look at is to see if PTEN-associated molecules, such as mTOR, MAPK, if they are affected by the intervention, and whether the changes in these molecules is correlated with clinical change. What is everolimus? It's an oral derivative of rapamycin. It's been in clinical development since 1996. As Dr. Eng mentioned, it's been approved for a lot of cancer. One is renal cell cancer, was in 2009, pancreatic neuroendocrine tumors, breast cancer, immunosuppression after organ transplantation, and recently for subependymal giant cell astrocytoma and tuberous sclerosis. In PTEN, the mTOR pathway is abnormally upregulated. It's very active. And everolimus or medication or compounds similar to PTEN, they inhibit this pathway. So you have, imagine like a car that's going very fast, and medication like everolimus will slow it down to hopefully normalize the speed of the specific pathway. This is a trial overview. It's a phase 2 trial. It's not a phase 1 trial, because usually phase 1 trial is looking at toxicity in healthy volunteers. So this is the first study in individuals who have a specific disorder, in this case, PTEN mutation. It's a double blind, which means the parents and the family does not know if the patient is on the active or on the placebo, and also the investigators, they don't know if the patient is receiving the drug or the placebo. It's a multi-site trial. We'll talk a little bit about it. And I mentioned the primary endpoint is safety, and then comes effect on neurocognition. These are the trials, the location of the trial. We are Boston children. You cannot see that. Let's see if we can. There we go. This is Boston here, Cleveland, and Stanford on the West Coast. So anyway, to cover a little bit the whole country, there were discussions about having an international site, but since this is our first trial, our first collaboration, it was a little bit challenging to bring in an international site in the beginning. This is the clinical trial organization. You have the 3 sites with Boston, Stanford, and Cleveland. And we look at this slide, you realize that it definitely takes a village to be able to implement a trial like this, a very complex trial in a rare disease. The funding, we have funding from NIH, Novartis, PTEN Research Foundation. The Boston is functioning like the main site with the coordination. They have the neuropsych coordination center. They will be processing the resting EEG and the auto-evoked potential, and the biostatistician actually will have support from Boston, but as well as from a biostatistician at Stanford. Cleveland will be responsible for the microbiome sample collection and the biorepository for all the blood samples. Let's talk a little bit about an overview of the trial design in terms of participant flow, the inclusion-exclusion criteria, study design, endpoints, and the efficacy composite score. So as Dr. Eng mentioned, we're looking at recruiting and randomizing 40 individuals. 20 will go into the active arm when we will be receiving everolimus for 6 months, and at the end of the 6 months, they will exit the study. In comparison, 20 individuals will be randomized to the placebo arm for 6 months, and at the end of the 6 months, the individual who received the placebo will be offered to participate in an open label phase when they will be able to get everolimus openly. The goal is to recruit 10 at Stanford, 10 participants at Stanford, 10 at Boston Children's, and 20 at Cleveland. Now, the inclusion-exclusion criteria, this might be boring or might put some people to sleep, but these are very important for several things. One is for the safety of the participant, to make sure that the participants who are included in the study are healthy. Second is to make sure that the study can be replicated by others when it's completed. So the age, originally we started was the goal of recruiting individuals between the age of 6 and 21, and now we're in the process of expanding this age range to include kids as young as 5 all the way to adults to age 45. And you have definitely, they have to have a confirmation of a PTEN mutation. The third inclusion criterion is to have a non-verbal IQ or an IQ of 50